I'd like to welcome Dominic and all of you to the live question and answer session of the seminar. Please ask your questions in the box on the screen and we will ask the most popular questions. So if there's one you'd like to see answered, please do use the upvoting function. Thank you so much for your talk, Dominic. And the first question asks, how has COVID-19 impacted efforts to control malaria? And also whether the supply chains being established for coronavirus vaccination, could they be used also to help malaria interventions?
Great question. Well, one of the biggest things in malaria control is distributing insecticide-treated bed nets to Africa, and also making sure that people who've got malaria have effective treatment. So anything that affects those logistics chains is going to be problematic. And we know in the past, for example, in the Ebola epidemic, we know that that had big impact on malaria control in the region. On this occasion, there's been a lot of effort by the major funders, the major stakeholders, to try to mitigate the risk of that and put in place processes that mean that those things aren't disrupted as much as they would have been. There undoubtedly has been an impact, but there's a lot of consciousness of the need to keep normal supply chains going. So I think the answer, I think the jury's out on that, actually. I think in terms of supply chains, it's interesting, it might almost be the other way around. A lot of, although malaria control has its challenges, it does have quite strong infrastructure in many countries. And I think that many of the systems set up for malaria control will actually be quite helpful in delivering widespread vaccines. And at a more technical level, I can proudly say that one of the groups we've been helping to get in-country genome sequencing going for malaria in Ghana, our colleagues in Ghana, they were I think the first African group to sequence COVID. So it can work the other way around as well. And one of our sort of mottos for the future is, if you could put in place robust systems for surveillance and treatment and prevention of endemic diseases like malaria, it's a way of preparing for epidemics.
That's brilliant, thank you.
Great, thanks Dominic. And thanks to everyone who's voting. It's really helpful to see questions popping up the list. So I'm going to go to the next most popular one. So Dominic, how will climate change affect the distribution of malaria? And will the UK actually begin to show infection transmission?
And not just the UK, I mean, what about Cambridge and the Fens? Because actually, when there used to be malaria, it was all in the area around the Sanger Institute. Yeah. Well, I mean, there's no question that the mosquitoes that transmit malaria, Anopheles mosquitoes, do like warm, humid conditions. And there's no doubt that if the world heats up and levels of water rise, that will undoubtedly see a proliferation of mosquito species. There's no doubt about that. On the other hand, in fact, a lot of the mosquitoes, I mean, there are some great mosquito species that can transmit malaria in North America right now. There are some very strong malaria transmitting mosquitoes. It's just that in North America, because they've managed to get rid of parasites so effectively from the population by multiple mechanisms, there's not malaria endemic in those countries. So I think the answer is the risk will increase, but I think as long as we can tackle the parasite now, it may be we mitigate the risk and prevent it spreading. So I think now is the time to act at multiple levels.
Okay, and is that, you know, you kind of touched on in your talk, is that primarily by reducing marshes and stagnant water? Is that how those, you know, in North America, they've been able to effectively control it despite the fact that there's prevalence there?
That's a great question. I mean, it's you have to throw everything at it. Preventing mosquitoes breeding is a great start, but actually even in parts of North America, that's not sustainable as a single mechanism. But if in addition to that, you treat everyone who's infected as promptly as possible and get mass treatment going down, and you drive the mosquito population down. And also in terms of, you know, living conditions, if they happen to be indoor biting mosquitoes, if people live in houses that don't have mosquitoes inside them, either by the use of bed nets or other mechanisms, all of that drives things down. And in that sense, you could think of it as being a bit like COVID. You're just throwing the book at it. You know, if you've got a vaccine, you use a vaccine, but you also have to use other sensible measures, you know, that may involve, you know, things that can prevent transmission. But But yes, there's no question that in the early 20th century, engineers had a great deal to do with prevention of malaria by essentially building systems that improve drainage and so on.
Great, thank you.
Thanks to all of you upvoting questions. We've got the popular one at the moment, Dominic, is what is your proudest output from malaria, Jen?
All right, thank you. There's lots of them, but I think it's mostly about people, and I couldn't go through the long list of people, but I will just say that we invested very early on in training capacity building in malaria endemic countries, particularly in Africa. And we're very proud that many of the people who were just learning about genetics as we started off are now leading large programs in Africa. And importantly, not just leading large research programs, but leading large capacity building programs and teaching themselves, and now helping to build the next generation of PhD students and postdocs in Africa. So that is certainly a very proud thing for us.
Dominic, one of the top ranked questions is, how optimistic are you about the potential of the next gen of antimalarials that target conserved loci and so are less likely to drive resistance?
Oh, heck. Well, okay, so sorry, was that about antimalarial drugs?
Yeah, the next generation of antimalarial drugs that target conserved loci and then potentially reduce resistance mechanisms.
Yeah, okay, well, I think there's two parts to that question. I mean, well, multiple parts to that question. Firstly, just finding any antimalarial drug at all is jolly difficult. And although we have been blessed by some absolutely fantastic drugs, I mentioned 3 in my talk, and there's a bunch of others, it is also true to say that finding a new one is extraordinarily difficult. In fact, trying to find new antimalarials was one of the largest military spending projects during the Second World War, and they didn't come up with that many. Fortunately, you know, anyway, so just finding any drug is difficult. And actually, I would say the work going on at Sanger and elsewhere is very much targeted on screening candidates and looking for new targets. So I think the question of a conserved region of the genome, I'll give you my take on that. As far as we know, every region of the genome is mutating all the time in a few individuals. And if we go back to the talk we had from Peter Campbell about cancer, it's very similar. Just as somatic mutations are happening in cancer all the time, mutations are cropping up all the time in malaria parasites. And I don't think there's, in my view, as far as I know, there are very few regions of the genome, if any, that have no mutations. Now, there are some that tend to be more conserved because there are selective pressures on them that actually lead to them being conserved. And actually, interestingly, the Kelch gene, which is a gene that's involved in artemisinin resistance, turns out to be a highly conserved gene evolutionarily. That hasn't stopped it acquiring a lot of mutations under the pressure of artemisinin resistance. So I think it's all a question then of evolutionary balance. A gene may be conserved because there's been some historical reason for its conservation across species. But if a parasite is now forced, sort of live or die decision, those mutations may come out. So I think it would be phenomenal if there was a drug that did the thing that has been asked about, but I can't see that happening immediately.
Thank you.
And we have a question from Sophie, who asks, how are you accounting for the chance that missed sampling might demonstrate insecticide resistance arising earlier or concurrently in multiple places? I'm perhaps being a bit dim. I haven't got the full, perhaps I haven't got all the meaning of the question, but let me have a go at it. I hope, Sophie, I get this right. So I think what you may be asking about is insecticide resistance happening all over the place, and are we finding it all? And the answer is undoubtedly it is happening all over the place, and undoubtedly there's many types of insecticide resistance we haven't seen. That being said, what we can see in the populations we've sampled across a wide section of Africa is there do seem to be a few dominant types, and obviously
the more intensely we sample, the greater chance we have of picking up the whole picture. And an important project that I didn't actually mention during my talk is working with the Pan-African Mosquito Control Association. It's an African-led population of entomologists working across Africa on mosquito control. And we're working with them. They're collecting samples from many different parts of Africa. So we're trying to get as comprehensive a coverage as we can so that we do pick up all the variants that are out there.
I think that was exactly the right the question. You answered the correct question.
Thanks.
Thank you.
Great. Dominic, you've got a question from Thanat. What could be the possible mechanism or mechanisms underlying the flow of pyrethroid resistance mutations between sibling species?
Well, that's a great question.
Maybe you could have just explained a little bit the question.
Yeah,
to us
I will.
well.
Yeah. I think that. Okay, so let's just pull that apart. So pyrethroids are the most commonly used insecticides against malaria right now, for a number of reasons. One is they've been very effective. Secondly, they're derived from natural compounds. Pyrethroids themselves are synthetic, but they're quite close to natural compounds, so possibly a little less damaging than other insecticides out there. And another is that they have some great formulations have been used to allow to impregnate bed nets with them. So you get these long-lasting nets. And so hundreds of millions of these are distributed every year. So we know that that is the single biggest tool we've got against malaria in Africa. And so the question is now the mosquitoes are getting resistant to pyrethroids increasingly. What's been found though, and there are multiple independent origins of insecticide resistance. One of the sort of worrying and puzzling things is that if a particular species picks up that insecticide resistance, you can sometimes find that in another species, and you know it must have crossed that species barrier, because when you look at the genome sequence, the underlying haplotypes, it's clear that that mutation was happening on a haplotype that must have been transferred from one species to the other. And what that brings out is something actually we've known about Anopheles populations for some time, that they can have very porous species boundaries. There are many different species of Anopheles that transmit malaria. Some are very distantly related, so there's rather little chance of genes transferring, but quite a few of them are quite closely related. And the best examples of the thing that's being asked about in the question is between mosquitoes that are members of a so-called Anopheles gambiae species complex. These are mosquitoes that clearly are different species on one hand, but they are a part of a sort of sibling complex. And it's fascinating that they do hybridize. You do get fertile hybrid offspring. And so that's how the simple answer to the question is, it's because the species can mate and they can lead to fertile offspring. So the question actually then becomes, well, why are they still species if that can happen? And there's this bizarre thing going on that they tend to stay as different species, although they can clearly transfer genes between them. And we think in that case that what's causing them to speciate are sort of ecological and behavioral things that make it favorable them to choose one species or the other species. But there are probably multiple mechanisms going on.
Interesting, fascinating, thank you.
We've got a question here that asks, what could your colleagues working as scientists in malaria endemic regions teach us as scientists working in COVID and endemic times? What can we learn?
That's a great question. Well, an awful lot. I mean, I think the first thing they could teach us is, what it feels like to be challenged by an infectious disease. I mean, we take a lot for granted, don't we? We're a rather entitled group, and so living under these sort of battle conditions is something that in malaria endemic countries that happens all the time. I think that I don't have a mature answer to that question. I'd like to think about it, but just off the cuff, it does go back to something I was saying earlier, that public health systems for dealing with endemic diseases in many malaria endemic countries, they're challenged, but there are structures for it. We sort of there is an understanding of how you can, on a large scale, deliver public health. And of course, we do that in the UK, but in a very different context. And I think a key issue there is often resource allocation. With limited resources, how do you target your resources for maximum effort? And actually, that's what we found at the beginning of the COVID epidemic, wasn't it? That, you know, we sort of found that we didn't have full resources. We were running out of stuff, you know, and so how do you target things for maximum effect? I don't have a great answer to the question, Julia. That was just an attempt.
No, that's fantastic. And we might get you back in a few months to reflect on this.
I think we need a learning session. I think we should follow that up with a learning session.
And Dominic, one of the most popular questions here, I recognize is not your area of expertise, but I think given the power of the crowd, I'm going to ask it anyway. And there's a question of how do gene drives work, and what are the ethical issues associated with it? I know that, again, this isn't your area of expertise, but at least what are the issues themselves?
Well, okay, so gene drive is essentially it's a form of genetic modification, and for some years, people have been trying to modify mosquito populations, for example, to try to stop them carrying malaria so well. But the powerful intuition now behind gene drive is that if you take a selfish gene, and CRISPR-Cas is a great example of a selfish gene, and you insert a copy into the genome, it will then tend to copy itself into the other chromosome, and as such that will tend to spread through the population at a much higher rate than any normal gene. So you can drive genes into the population, and a great strategy that's being developed by Austin Burt and colleagues at Imperial College in the Target Malaria Project is to do that to a gene called double sex. It's a gene which is involved in sexual differentiation, and where having that gene in the male is okay, having that targeted disruption in the male is okay, but if you've got two copies of it in the, if a female is homozygous for that gene, they become sterile. So you get that to spread through the population. Now, that's a great way potentially of driving down a mosquito population. So what's the ethical issues? Well, the first issue is it right to extinguish a species? Well, you could sort of say that, you know, fair's fair. I mean, the mosquito's trying to extinguish us, and, you know, it's just a war, and we could do a trade-off, and we could, I think that if we had that debate, it might be that many people would feel that was just fair. We're being challenged by this species as an enemy. We should go for it. So that debate needs to happen. Is it right to extinguish a species? People to take a view on it. Let's just assume people thought that was okay. There's another question, which is it safe? And it might be unsafe if it spreads to other species. Now, you know, in that debate, you could say, well, there's actually thousands of Anopheles mosquitoes out there. If we just target one, that's not a big loss. That won't affect the ecosystem. The trouble is that because of species complexes that I've just been mentioning to you, exactly, you can get flow across sibling species. Where will it stop? You know, how, in other words, we think it's safe, but do we know everything? You know, could we have a calamity? And what if it now spread through the whole ecosystem? Gosh, that wouldn't be a good thing. So it's a question of some of those things are sort of moral discussions, others are sort of biological ones, and that's what the debate is about.
Okay. But they are still being developed nonetheless at the moment? Well,
absolutely. And in fact, there was a fantastic article in The Economist a few months ago about this whole issue. And basically the question is, you know, are gene drives good or bad? And I think this is just the view of The Economist, which you may or may not want to believe, but it was that on the whole, they're probably not a good thing. They are probably rather dangerous, but there are some specific applications where it does seem that the positives outweigh the negatives. And the one sort of, if you like, clear example of that people are going for is the Anopheles mosquitoes that transmit malaria in Africa. That seems to be both a valid, a feasible target, one that seems, as far as we can tell at the moment, to be relatively safe. But of course, there will need to be a lot of safety data around this. There'll need to be sort of, there are ongoing at the moment trials that are trying to assess the safety, how much it will spread. Are there other implications of spread beyond a single species and so on?
Fascinating. I think we better just conclude the question and answer session there. There was more questions, but I think it's obviously been fascinating to learn about the science, about the the socioeconomic impact of malaria, the human cost. So thank you so much. Very fascinating talk and question and answer session. So I'd just like to also remind the viewers out there that this is a monthly event. So please join us in a month's time when the next speaker will be Gausha Trinka on the 14th of December. And she is a faculty member in our human genetics program and also the experimental leads of the Open Targets Consortium. And she'll be talking about immune genomics and how human variation impacts on the immune system and autoimmune diseases. Just also a reminder that all of these seminars, as well as the question and answer sessions, are available to view through the Sanger website and through YouTube. So please have a look at them and share them with your friends if they would be interested. And thank you so much for joining us today, and we'll see you next month. Thank you again, Dominic.
Thanks.